Thank you, everyone, for coming to the Responsible Conduct of Research seminar, training, whatever word you want to use today. My name is Karen Humphrey. I am with Environmental Health and Safety, and we're going to talk a bit today about lab safety. Let me tell you a little bit first about EHS. As soon as I figure out how to advance the slides from here. Looks like that'll do it. All right, wonderful. We're currently located in a little temporary building behind the Student Life Center. So if you go down the road down behind the Student Life Center, you're going to see a satellite dish, and we're in the little portable building behind that. That's where we're located right now. But you're going to often find a lot of us roaming around the Baylor Sciences building or any of the other buildings that house laboratories, because that's what we do. Our senior director is Dennis Nolan. He couldn't be here today, so I am stepping in his stead. Again, my name is Karen Humphrey. I'm the Lab Safety Program Manager, the Radiation Safety Officer, the Laser Safety Officer, and other duties as assigned by boss. So I do a lot of different things. Wendy Clement, who you'll see in the second part of the presentation, she's our Biological Safety Officer. You also might have a reason to interact with Clinton George, who is our Environmental Manager, or Zach Jones, who is the Hazardous Waste Specialist. So those are the laboratory people. We have other people in the department as well that deal more with general campus safety and student safety and whatnot. As a department, EHS does several things. We provide training, more of the general overview training of safety and how to deal with things. We do periodic inspections, both on the chemical side, the biological side, radiological, etc. We do monitoring, as is required by certain regulatory agencies. If you work with, say, radiation, for instance, we're responsible also for the ultimate disposal of hazardous materials. And if you are involved in any shipping of hazardous materials, we may also be helping you out with that. And lastly, we do accident investigation, because occasionally things go wrong, and these become learning opportunities to see how we can improve moving forward. I want to start by talking about training, because training is a learned skill. You don't know what you don't know. So when you first come into a lab, you get trained about a lot of things. And this is a 30,000-foot training. All right, this is the overview training, and this is what EHS provides. When it gets down to the nitty-gritty training of what you are doing in your specific lab with your specific project, that's going to be more lab level training that is provided by your PI or supervisor. The training is based on the lab hazards. So when you enter a lab, you're going to, you'll be assigned job categories in our lab management software, which is called SciShield. And that's going to determine what trainings you get, so that you don't have to take anything that is not involved with the work that you're doing. Most of our trainings, a high percentage, are one-offs. You train when you start your project, you're done, you don't have to worry about it again. There are certain trainings that require refreshers, because these are the things that are more apropos to what you're doing, more hazardous, more rigorous. And so those require periodic refreshers to make sure that you stay on your game as you're doing your work. If there's any sort of training that you don't have, there's a possibility that's something we need to add. So feel free to reach out to EHS and say, hey, we got a lot of people doing this. Is there a training available for that? And maybe there is. We can put it on SciShield and you are good to go. One of the other things we do are inspections, and there are 3 levels of inspections that we do. The general EHS inspections. If you work with lab hazards, you're going to see me once a year, maybe twice if you're particularly hazardous. You're going to see Wendy, the biosafety officer, once a year for the biological inspections. And if you work with anything that's regulated, you'll see us for different inspections for that. In addition to that, in SciShield, you have what's called a self-evaluation. Twice a year, once every 6 months for the chemical side, you go in and you do a self-evaluation. The cool thing about the self-evaluation is it's like the pre-test. Everything on the self-evaluation mirrors what I look for in my inspection. So if you're good there, you're going to be good whenever it is that I come into your lab to look around. We do a lot of things based on regulatory agencies, and we have several regulatory agencies that dictate why we do what we do on the federal, state, and local level. The big one on the federal level is OSHA, the Occupational Safety and Health Administration. They're responsible for hazard communication, for the lab standard, for the respiratory protection, standard for a number of different standards that all come together into what we do. And we do it so you don't have to worry about what all those little standards say. We just sort of distill it down. Here's the things you need to do. You do them, and everything's good. Another one that we interface with is the Environmental Protection Agency, and this is the Hazardous Waste Materials, the Clean Water Act, the Clean Air Act, to make sure that what we're doing is not impacting the larger environment in any sort of detrimental way. We also work with the Department of Transportation. Any hazardous materials that get shipped out go on roads, and that has to be done appropriately. If you are shipping out any sort of samples or anything from one place to another, again, they're going on roads. So there are certain regulations that have to be followed there. On the state level, primarily we deal with the TCEQ and the Department of State Health Services. Now, the TCEQ is the environmental quality. That's the state version of the EPA. So any sort of releases into the environment, any sort of hazardous materials, this all has to get reported there as well. The Texas Department of State Health Services, this is focused on any radiation work or any laser work that we might be doing. We have certain regulations we have to abide by there. And lastly, on the local level, we have the city of Waco. They have their own regulations in terms of what we can put down the drain, what we can maybe put in certain other places that are maybe not so applicable to labs, but in general, campus-wide sort of things. Those are the general things that will apply to most labs. Now, some labs might be working with controlled substances. And this is a drug enforcement agency. Anytime you're working with any precursors to illicit substances or illicit substances themselves, narcotics, anything that is considered a controlled substance, you're going to come under the purview of the DEA. And the DEA says you have to have a license to work with that. And not only do you need to have a license, everybody on that license is going to have had a background check. And because you have that license, now you have to do certain security and inventory things to make sure that those controlled substances stay controlled and don't get diverted to do something that they're not meant to do. Anybody that has a DEA license, basically the DEA can walk in, surprise, we're inspecting you anytime they want to, because you are a license holder. If that were to happen, call EHS, because the last thing we want is for any of our PIs to be going through an inspection alone without a sort of backup from us. Another thing that is regulated similarly to a controlled substance is tax-free alcohol. Some labs work with 100% alcohol, it's drinkable, okay? Now, if you pay taxes on that alcohol, no problem. If it's tax-free, then there's this little regulation by the alcohol, tobacco, and firearms place that says that you have to treat that as a controlled substance. So you need an inventory, you need to keep it locked up. Again, this is for purposes of not diverting it, but it's because it's tax-free. So it's really sort of interesting. It's not a safety issue, it's the government wants their taxes on an issue. So that's another thing to be aware of. If you work with radiation, now there are two types of radiation you might be working with. There are radioactive materials like any of the radioisotopes, or radiation producing machines like X-rays. We have licenses for both of those. We have a license for the radioactive materials. We have a state registration for the radiation machines. There are a lot of things in common between those two in terms of monitoring and being on the license, documentation, inventory, and things like that. Also, the state requires an annual audit. So you might see me for a chemical side, and then you might see me another time of year coming to look at your radiation side, okay? Because we need to make sure that those are documented and that everything is fine, because we get inspected by the State Department of State Health Services. It's about every 2 years for the radioactive materials program, and about every 3 to 5 years for the machines program. And we'll know in advance of those. So it's not quite like the DEA where they just sort of knock on your door and say hi. We will have some advance notice, and we'll be able to help you out to make sure that everything is ready to go there. If you work with lasers, it's similar. We have a state registration for lasers. And it's not quite as rigorous as for radioactive material, but there are still some things in common in terms of signage, in terms of making sure we have appropriate warning labels, appropriate training. Again, another annual audit of the program is required by the Texas Department of State Health Services. So if you work with lasers, again, you might see me on the chemical side at one point in the year, and again for the laser side during another point of the year. Going back to chemical lab safety, there are two regulations that kind of work together for chemical lab safety. One is OSHA's hazard communication standard that says that as a worker with the materials, you need to know what you're working with and how it can hurt you. So these are where things like labeling and safety data sheets and whatnot come in. But HAZCOM is older, and it was originally developed more for industry. So people that are working in large volumes, but they're not working with that many chemicals. In laboratories, we can work with scads of chemicals. And OSHA finally figured out that HAZCOM is okay for labs, but really there's a little bit of a better fit for laboratories. And so they came up in the 90s was what's called the laboratory standard. And you can see that there's overlap between the two of them. You know, they both require labeling and material safety data sheets and training. The lab standard puts a little bit more paperwork in there in terms of the chemical hygiene plan, standard operating procedures and whatnot. Now, what we do here at Baylor is we sort of married the two, all right? In that you have all of the requirements and lab standards, plus we keep an inventory of all of our materials. And we did that for OSHA, but we also do that for the local fire department so that if they have to respond, they know what it is they're responding to. You might see a couple of different types of labeling. The older style is this one right here. This is the old NFPA, still used in a lot of places, particularly on like storage areas. So you're still going to see this around campus. More so in laboratories, we've gone to what's called the GHS standard, the globally harmonized system, which is better fit for lab, because what this was based on was the highest hazard in the lab. So it was pretty common to go by a lab and see all fours, which is the highest hazard, plus a lot of things here. But the thing that made it a 4 might be about that big. So it's really not that informative. This is a little bit more informative because now it tells you what those things are. And it's not just telling you you have a health hazard of 4, it's telling you, oh, we've got flammable liquids, or we've got something that's corrosive, something that's acutely toxic, and things like that. So this is primarily what you're going to be seeing a lot of is these pictograms, right? And you're going to get way more information about this when you dig into SciShield and you start doing that training. This is just the 60,000 foot view before you get 30,000 foot view in SciShield, right? On laboratories, what we do is we have a sign that has a lot of, you saw those in the last slide, a lot of these listed to let folks know what's in there. Not only people who are coming into the lab, but first responders that might be coming into the lab at some point. What your PPE is in the lab so that you know the moment you walk in what you got to put on, and any sort of weird regulatory stuff like lasers might be here, radiation might be here. We don't put controlled substances up here because we don't want the world to know we've got controlled substances in our lab. But you're going to see this in front of every lab. It's going to have contact information, going to have our contact information for EHS, a barcode if you have access to SciShield and want to find out more information. This should be required annually, or should be updated annually, because people come and go in labs, and we need to know the most current information. Safety data sheets. There used to be a time when we had binders and binders and binders of safety data sheets in paper, in the labs. It was an administrative burden for people to keep up with. Thanks to the interwebs and everything at our fingertips, we no longer have to do that. There are scads of ways you can look up a safety data sheet anytime you need it. The SciShield homepage has it. Manufacturers' websites, for instance, Fisher VWR, are clearinghouses for those as well. You can Google a safety data sheet and come up with a million different versions of them. If the power goes out and for some reason you need a safety data sheet, call EHS, call us at EHS, and we will get them. Get them for you. One place where you will need paper is if there's an emergency and you have to send somebody to urgent care or the hospital, print a copy of it and send it with a person. Yes, the hospital has the ability, same as we do, to call up a search engine and search the safety data sheet, but you're going to save them some time if they walk through the door and hand the responders the safety data sheet for the chemical. Another piece of paperwork that we have in the lab is called the chemical hygiene plan, and this is sort of the lab overview, if you will. This is the control measures that are in the lab. You know, we have fume hoods, we have biosafety cabinets, we have this, we have that. What personal protective equipment is required of you in the lab, what to do if something goes wrong. Here's how we in this lab acquire our safety data sheets. Here are the standard operating procedures. Here's who to contact. All of that information is in there. Luckily, it's condensed. So the chemical hygiene plans are generally 4 to 6 pages long. So it's not a huge tome that you're looking at. We also require a chemical inventory for all of our chemicals. Now, it's redundant. In the Baylor Science Building, it's easy because all the chemicals come to the stock room. The stock room puts the barcode on it, sends it up to the lab. If you're not in the Baylor Science Building, well, you don't have a centralized reception place. It's probably coming directly to your lab, in which case it's up to you to figure out, how am I going to keep an inventory of these chemicals? So they need to come in, and also they need to come out. And the reason they need to come out is because our buildings have what is called maximum allowable quantities. This is the most amount of flammables or toxics or a few other categories that are allowed to be had in the building. And so we need to know an inventory so that we know, are we close to the maximum allowable quantity? Where are we with that? So it's really important to pull them out, because if you never pull them out in 10 years, everybody's over the maximum allowable quantity. So be sure that you're pulling them out as well as putting them in. We've started putting this as part of our inspection regime, so that one of the things I'm looking for is, do you have an inventory, and has that inventory been updated recently? Okay. There's a picture. I forgot that was there. This is a barcode that goes on Baylor Science Building chemicals, and maybe there can be a way to have barcodes in other buildings as well. Just reach out to EHS and see what we can do. Lab security is really important, okay? And it's important because there's glassware that can be used for illicit purposes. There are chemicals that can be used for illicit purposes. And so we need to make sure, and there are biologicals that need to be kept track of. So there are reasons why we must keep the lab locked. I know it's really kind of tempting to prop the door open when you run out to the bathroom. Don't do that. Lock the door, take your keys with you, right? If anybody is in the lab and you don't know who they are, challenge them. You know, you don't have to be rude about it. You say, hi, may I help you? If I'm in the lab and you don't know why I'm there, challenge me. I am not going to get offended. In fact, I'm going to say kudos, because you did what you were supposed to do. You saw somebody that didn't look right and you asked them, may I help you? Why are you here? If you see something suspicious, pick up the phone, 254-710-2222, call Baylor PD, let them know that something doesn't look quite right. Right? Security is really important. Let's shift gears just a little bit. Get a drink here. And talk about if you have to ship hazardous materials. This doesn't happen often, but it does happen occasionally. Now, because materials go on roads, there are lots of regulations from the Department of Transportation of what you can and cannot do, right? Some of that shipping information is going to be in your safety data sheet, but it's not all going to be there. The easiest way to do it is not do it yourself. Okay, DHL, UPS, they have people that are trained in shipping of hazmat. They will make sure that it is packaged correctly, that it has all the correct information, that it's done. If you do want to ship it yourself, the DOT training is required for anybody that's shipping hazmat. That's about an 8-hour training. Or you can reach out to EHS and see if we can help you. In a lot of cases we can. In some cases we may not be able to provide as much help as you would like, because we're not DHL, we're not FedEx, we're not UPS. So Just make sure that you're not, just slapping a label on it, sending it out through the post office, because they're gonna find it. If it goes by air, they're gonna check it at the airport. You're gonna lose your samples or your materials. It's not gonna be nice. You might get fined. It's just a whole lot of trouble that you don't want to bring on yourself. So be aware of that if you should want to ship any hazmat. If you want to transport it yourself, don't use your personal vehicle. Okay. Again, there are DOT regulations. Use a Baylor vehicle. Use the appropriate hazmat packaging. Put, of course, the SDS with it. Don't carry it in your luggage, because it will get found when you're screening the luggage, and it will get confiscated, and you may get in a lot of trouble. Just a few tips there. Now, what do you do with the stuff you make in the lab that you want to get rid of? Well, this is chemical waste, and we have a nice little form. Make sure you're using this form, by the way. You'll know it's our most current form because it's got a date right there. This one has more information and instructions than previous forms have. So make sure you're using this one. Don't put the date on it. And the reason we ask you not to put the date on it is because the date starts a regulatory clock. But that regulatory clock doesn't actually start in the lab. It starts when it gets removed from the lab down to our central accumulation area on the first floor. So don't date it. That's for us to do, not for you guys. Don't mix your wastes. Those of you in established labs, you've got your waste streams set up. Use those. If you're in a new lab, reach out to us. We can help you figure out what your waste streams need to be. Use the appropriate containers. Some things will eat right through plastic. Some things don't like glass. Some things, you just have to sort of know what you're working with. And again, we can help you figure that out if you don't already know. Very important to keep them closed when you're not actively adding stuff, right? That's an EPA violation. We've actually been nicked by that in the past. So make sure that you are keeping your containers closed. As far as the waste storage goes, in any lab, you're limited to 55 gallons, okay, which is a lot of you might say. And then for some labs, they'll make that 2-3 times a week. So just, be aware of how much you are accumulating. Set up one area in the lab to put your waste. This makes it easier for the people that are coming in to grab it, so they don't have to go, where's your waste, and go around to place to place to place while you're trying to work in the lab. So set apart a place for it. And have a spill kit, not just for the chemical waste, but also for any chemicals you're working with, so that should they spill, you know what to do with them. Don't pour anything down the drain unless you have EHS approval. Also, don't move it from the room that it's generated in to another room. That's our job. There's like training. You have to be certified in order to do that. No one in the lab is really certified to do that. So once you make it, it stays there until somebody from EHS comes to get it. On the website, there's a waste form, pickup form. Fill that out when you are ready. Don't let the waste accumulate, all right? Because then you're putting in a big pickup form, and they may not be able to come get your waste, because we have essentially a size of a large walk-in closet to serve the entire university. So if you are putting in a bunch of waste, and this person is putting in a bunch of waste, and this putting in a bunch of waste, we don't have room for it. So it's better to do it a little and often than to do it once a semester and clean out all the waste, right? So just a word to the wise there. Make sure that whatever is on the paper form is matching what it is that you're putting on the pickup form, because that pickup form becomes a regulatory document at the end of the day that gets handed to the waste people. So everything has to match up, or there's going to be some issues. That concludes my part of it. So I'm going to hand it over to Wendy now, who's going to take me through some biosafety. All yours.
All right, thank you, Karen. My name is Wendy Clement. I'm the biosafety officer here at Baylor University. Just real quickly, some of the responsibilities that we have in EHS biosafety that I oversee is the IBC, Institutional Biosafety Committee. We do biological risk assessments, protocol summaries. We coordinate with researchers that are starting new research, or maybe amending current research if they're including a new material or a new technique or protocol or whatever. We also deal with biological and medical waste, particularly like animal specimens and materials such as that. We can get rid of those for you. Most of the biological waste that's accumulated in many labs have their own autoclaves within the labs themselves that are being monitored and maintained by the lab. We will pick up sharps for the most part and remove them. And again, like I said, any PPE or animal waste that usually gets discarded by EHS either throughout the semester or at the end of a school term. We also are in charge of the biosafety cabinet and chemical fume hood annual certification. If you are coming into a new laboratory and you're bringing in new equipment, you contact EHS. I will set up an outside vendor, come in and get your equipment certified. If you're moving from one space to another space, or you're having to renovate, or something's happening with your biosafety cabinet, contact me so that we can get you set up as soon as you're moved over. We'll bring a vendor in, they'll certify your biological safety cabinet and you're set for the next year. Finally, we are starting to look into coordinating with PIs and students on field research. So if you are going abroad and you're collecting samples, please talk to us before you go. We don't need you bringing your biological samples back in your luggage. We don't need you bringing your samples back, even if they're shipped and you don't have appropriate signage, permits, paperwork, depending on where they're coming from and what they are. So touch base with us at EHS. We can help coordinate. We can make sure that you've got appropriate paperwork in place and that you are ready to go and you won't get hung up in customs and lose your samples. Some of the biosafety guidelines that we live and breathe by, if you're working with recombinant synthetic DNA, and of course the NIH guidelines, they were most recently updated in 2019. We are anxiously awaiting for the next iteration of the NIH guidelines that in 2019 had a lot of gray areas, particularly with utilization of new techniques like CRISPR and certain genetic modifications that were not clearly defined. And so hopefully the ones that come out maybe this year or early next year will be a little bit more specific so that universities that receive funding from the National Institute of Health can abide by these, because if you are getting funding, you have to follow the guidelines if you're utilizing recombinant synthetic nucleic acids. The second thing is the BMBL. That's the Biosafety Microbiology Medical Laboratories Handbook. It's now up to, I want to say 800 pages. It's very in-depth. The most recent iteration came out, I want to say in 2022, maybe the beginning of 2023. It has everything in there. So if you are working with biological materials and you need a copy of the BMBL, please reach out to me. I have numerous copies I can share with you, or if you have questions, we can go through them. There's checklists, there's all sorts of stuff regarding containment, training, PPE, the whole 9 yards within the BMBL. Finally, there's the OSHA bloodborne pathogen standard. If you're working with bloodborne materials, blood, tissues, cell lines, anything like that, this is going to fall under the bloodborne pathogen standard. Training for this is annual and is dictated by OSHA. So if you check in SciShield that you are going to be working with one of these materials, you will get pinged for OSHA training on the bloodborne pathogen standard every single year. I also have to take it every single year. So we are all in the same boat, and that is nothing we can really get away from until the standard changes, which I don't see it happening anytime in my lifetime. The Institutional Biosafety Committee is now housed in Environmental Health and Safety. If you're utilizing any of these materials, you're using pathogens, you're using recombinant synthetic nucleic acids, you're using biological toxins, doesn't matter the quantity. If you're working with human or animal tissues, please reach out to me because I can help you walk through the registration process in SciShield. Previous to this year, there were fillable Word documents and fortunately, the process has changed, hopefully make it a little bit easier now that everything is within SciShield, which you should already be familiar with if you're working in a laboratory. I have developed these little quick guides, which that's kind of an oxymoron because it's not quick, but it is a guide. So hopefully it can get you through the process. And like I said, I'm pretty easily accessible. Most everyone knows how to get a hold of me and I can help walk you through the process. Biological hazards that we're really looking for, and you should have filled out in SciShield is, are you working with anything that generates aerosols? Doesn't matter if you're working with nanoparticles, if you're working with biological agents, whatever you're doing, if you're creating a aerosol generating technique, you want to make sure you've got the proper training and PPE and engineering controls in order to meet those needs. If you're working with sharps, this is where the bloodborne pathogen standard comes in. We need to make sure we've got you appropriate first aid or processes in place in the event that you have an exposure, a bite, a cut, or a needle stick. Right now, we are not working with high concentrations or large volumes of certain biological agents, but this would fall under this category of biological hazards if you were utilizing it. And of course, the bloodborne pathogens as well. There's 4 biological safety levels, which is identified and described in the BMBL. Right now, Baylor University only has BSL-1 and BSL-2 labs. It will go all the way up to BSL-3 and BSL-4 labs. BSL-3 labs are laboratories that work with higher risk agents that do not have effective medical interventions for those. And then of course, the BSL-4 laboratories are the highest risk biological agents that have zero human recourse as far as medical interventions. And so those are the hemorrhagic fevers and some stuff that you might see in Hollywood producing type agents and the moon suits, the whole 9 yards. We do have separate designations for biosafety level containment for plants, animals, arthropods. Right now, our vivarium has ABSL-1, animal biosafety level 1. We have a room that is capable for animal biosafety level 2 if you're wanting to expose your animals to viral vectors, which we have some researchers that do that, or you're exposing them to some kind of biological agent and evaluating them for a set amount of time. That room has hand washing capabilities, biosafety cabinets, and most of the research is contained within that space. We also have arthropod containment here at Baylor University, both ACL-1 and ACL-2. Arthropod containment has its own designation, set of regulations and recommendations, and you would work directly with me if we're going to introduce any biological agents to our arthropods, or you're wanting to work with exotics that are not indigenous to the area. So those will also probably require permits, APHIS permits as well. So again, anytime you're wanting to work within any of these biosafety levels, please reach out and we can work together to make sure you've got everything in place that you need. Common BSL-2 agents, human-derived materials. We have a lot of researchers that are working with cell lines here on campus, blood, saliva, unfixed tissue, stuff like that. Those, I get risk assessments, we get PPE training in place, you're good to go. Some other things are viral vector constructs. We've got some researchers that are also working with viral vectors in various applications in conjunction with animal work. So again, you want to coordinate with EHS, me particularly, make sure we've got everything in place prior to initiation of work, which may include application to the IBC as well as application to IACUC before you start work to ensure that all of the regulatory compliance is addressed prior to initiating research. More BSL-2 agents, we've got pathogenic microorganisms. Here we've got certain viruses that are being utilized. If you're working with Cryptera giardia, if you're collecting environmental samples, if you are out in the field and you're collecting raw sewage samples for your environmental class, or if you're in the field and you're collecting live mosquito studies, all of these biological samples are coming back to the university. You need to coordinate, make sure we've got risk assessments in place, you've got proper training in place, we've got proper containment in place, we've got proper shipping and receiving in place, and we've got any permits either on the local level, state level, or national level that may need to be in place prior to starting your research. Risk assessments, this is the cornerstone of all biosafety, which goes hand in hand with it depends. So I will commonly get researchers or students calling me and say, Wendy, can we do this here? And they explain the project and I go, well, it depends. How are you gonna collect the samples? How are we gonna transport the samples? How are we gonna manipulate the samples? What are they gonna do with them? We go above and beyond to try to accommodate researchers to make sure that science continues to move forward. We just want to make sure we've got everything in place beforehand to make sure that our students and our researchers are operating and manipulating the material safely. Some of the questions that we ask on risk assessments, and this is usually in conversations, even after I get the materials, like, what are you using? How are you going to manipulate it? What are you going to, how are you going to transmit it? Is this biological material transmissible? Do we need vaccinations? Again, like if you are handling raw wastewater samples, the people that are working in the labs should have their hepatitis series vaccines prior to initiation of research. If you don't have that, you need to reach out to us and make sure we can get you the appropriate information to go and get that initiated. If you are going abroad, you know, what is something that you might likely run into? Do you need to get rabies or look into getting rabies? So there's a whole host of risk associated information that you may need to know prior to collecting samples, or in the event you are exposed while out collecting samples. This is a hierarchy of controls, starting with elimination, substitution, engineering controls, administrative controls, and then finally PPE. So a lot of people think that if I slap on a mask, throw on some gloves, I am safe. No, this is the last resort. If you're wanting to work with material and it is a high risk material, can we substitute it or eliminate it? Do we have to have that material? If you have to have it, can we substitute it for something maybe a little bit lower risk that we can manipulate here on campus or within the whatever containment is available for your lab or available to your lab? The next thing would be engineering controls. If you are working in a lab with a BSL-2 laboratory, do you have a biosafety cabinet? Is your biosafety cabinet up to date and certified? Do you have appropriate airflow in there? Oftentimes you're in a laboratory, but the directional airflow is not negative, it's positive. I don't want you working with aerosolized agents that could then waft back into a research hallway and expose potential folks coming in and out. So there's numerous engineering controls that your laboratory should or may look into getting and putting in place equipment plus the actual structure itself before you even get down to the SOPs and the training, which is administrative controls. Do you have somebody that wants to work with whatever they want to work with, viral vectors? And okay, come on in. Are you using first generation viral vectors? Or are you using third generation viral vectors? All of those are, the risk goes down with the generation of viral vectors. Oh, okay, you've got all this in place. How are you, what's your SOP? Are you mouth pipetting? Or are you, you know, the SOPs are in place to help the student and help us at EHS understand how you're manipulating the materials. The individuals that are conducting those experiments and doing this technique have to provide those SOPs. I can provide you an SOP, but I'm not the one doing it. It would be like me telling you, writing an SOP on how to shower. You know, I'm not gonna ask you to start at your feet and work your way up if you're using a washcloth. You want to start at the top and work your way down. So SOPs are kind of the same way. You want the person that's actually doing that technique to write the SOP and provide the information so that we can make sure that it's being done correctly and efficiently and lowers the risk as much as possible for your lab. And then, of course, finally, the PPE. We need to make sure do you have PPE? Do you have compatible gloves? You know, do you need respiratory? Do you need face shields? Do you need disposable coats? Do you need sleeves? Do you need none of this? It kind of depends. And then are you trained properly on how to remove gloves if you've had an exposure, if you're working in biosafety cabinet and stuff like that? So again, if you are relying on your PPE to keep you safe, we need to visit. Please contact me and we'll start back at the beginning with the risk assessments. Again, engineering controls within the labs. This is directional airflow, making sure you've got sinks, making sure you've got eye wash stations. Are they checked? When's the last time you have actually unscrewed your eye wash to see if there's a filter in there? Is the filter falling apart? Is the filter full of debris? I mean, these are all things within your laboratories that are meant to keep you safe, that you want to check frequently for the health and safety of your students that are working in the labs. If you're wearing a biological safety cabinet and you are aspirating something, make sure you've got a HEPA filter in line so that you're not sucking up anything you're aspirating into the house air. Just another step in keeping folks safe. Let's see, biological safety cabinets. There is a training in SciShield on how to use biosafety cabinet. If you need additional information, let me know. I lived in one for almost a decade. So if you're uncomfortable on how to utilize it or when to clean it, or, you know, something is not sounding right, please reach out. We are here to help as much as we possibly can. Administrative controls, again, in the hierarchy of controls. This is the second to last step right before your PPE. This is your training. Are the people who are working with the materials familiar with what they're working with? Do they have any medical issues that may need to be addressed prior to working with material? It happens sometimes, and you want to make sure those are in place. Are vaccinations required? Can we help in facilitating that if you're working with a certain material? Administrative controls, everyone knows these or should know these. You know, no eating, drinking in the labs. Make sure you're wearing appropriate PPE while you're in the lab. Do not wear the PPE outside of the lab. Have your hair up. You know, no makeup, no chapstick, no bubble gum. If you're working with sharps, please do not recap your sharps. Just dump them in the sharps container. We will come and get them. I don't know how many times I've walked into a lab and the sharps are recapped. Please don't do that. Every time you do it, you're increasing the risk of a needle sticking if you're working with recombinant synthetic materials. And then now we have to file a report with the NIH. So eliminate that possibility at all. Just throw them away and then calls will come pick them up. Biological waste. This is one of those things that Karen had touched on. As far as chemical waste, biological waste, same thing. You see sharps container. This is not my picture, but I had one very similar to this. It actually looked like a lovely bouquet of needles sticking out of a way overfilled sharps container from a location on campus that will be unnamed. But don't do this. You're exposing pretty much everyone to the possibility of a needle stick. We don't know what those needles were utilized for in some cases. So if you are working a BSL-2 laboratory, you got dozens of trash cans, have them covered when not in use. We don't need anything being dropped in, aerosolized out incidentally or otherwise knocked over. If it could have happened. It did happen when I had my own laboratory. So, just go through and make sure you've got stuff covered. And then finally, the lovely picture of me down here in the bottom, picking up my biohazard boxes. I will come and remove, again, I've got roll-offs, 96-gallon roll-offs for animal materials. Usually they're going straight from the freezer into the 96-gallon roll-off and then out to the vendor. But we will also pick up certain bedding for vivarium if it's required. And it can't go out with regular bedding and stuff like that. PPE, this is a lovely picture of one of us, I won't say who, during COVID. Head to toe Tyvek, face shield, N95, gloves, shoe covers. This is above and beyond anything we'll ever use in our research lab here at Baylor. But this is an example of PPE that can be used and utilized when needed. This is more likely, what you see is one of our previous researchers. She's got coat on, she's got gloves on, she's got sleeve covers, working in a BSL-2 laboratory, actually working with a virus right there and a biosafety cabinet. So again, make sure your PPE is appropriate for what you're wanting to do. If you are utilizing N95s or you're utilizing PAPRs, please reach out to us. We have a respiratory protection and certification program that you need to be tested for prior to utilizing these respiratory protection units. And then finally, field appropriate PPE. It's going to depend on what you do. So if you're in the Arctic, you're going to have different PPE than if you're in the tropics. And don't get those mixed up because it could be a long trip. Incident reporting. In the event that you have been exposed, you dropped a vial of whatever you're working with, biological agent. You're working with applying viral vectors to your mice and you stick yourself. Whatever you are doing and you have either a near miss, you actually have an incident or whatever, you're going to add this to the incident report form. It's on the EHS website. You should be able to go in there. It's going to be, we have a new vendor, Campus Optics, that is going to be handling the incident report forms. You go in and populate them. Once you go in and populate the incident report form, it will go to someone in EHS. We will follow up, do an investigation, make sure you've got whatever needs medical or otherwise ensure that it doesn't happen again, or we can figure out what caused the incident and try to mitigate that for future activities. And then if there is a regulatory component, this is also utilized for a regulatory component. If we have to notify the NIH, if we have to notify EPA, if we have to notify somebody independent outside of Baylor, and we'll go from there. So on the EHS website, this is incident report form. This is where you go to report anything that might go wrong in the lab.